In order to make the plant cell, I have used this plastic cup from parcel boxes, and apart from the plastic cup, you also need the nail cutter's knife. You can also use a pen knife, but this is reliably, safer when compared to the pen knife. And just keep a tissue handy. I have just used already used a tissue, and I have just kept it aside. So just keep a tissue handy just to know. And obviously the clay. You can also use the clay of 50 rupees, which I used in animal cell preparation. Even that will be very great for this. So let's start the video. First of all, in order to make the plant cell, I'm using this surface so that I can clearly indicate the cell wall as well as the cell membrane. In order to first prepare the cell wall, I'm using the dark green one. And a cell wall in a plant cell generally is helpful for all the structural support, and it offers even the protection for the cell. And firstly, I just took a small ball of this dark green molding dough, and you just are going to do it like this. So after wrapping it like this, you're just going to place it around the first top wall like this. And in order to prepare the cell wall, I just took pieces of broken dark green molding dough. So the reason I'll tell you. So first of all, we are going to place this broken green molding dough beautifully like this, right? So in order to get it stuck little firmly, you need to press it. So I'm just for showing you for the video purpose. I'm not pressing it more firm because I'm going to reuse this clay for my other videos. Right, so here's my cell wall which is incomplete. So the reason here is cell wall generally in case of plant cells are made up of cellulose, complex carbohydrates like cellulose, hemicellulose and pectin. So in often they are generally accompanied by secondary cellular substances like the lignin, suberin and all, which are released by the cell wall, and generally because they are released by the cell wall, they present here anchored to this primary cell wall, right? So if you want to resemble them, I'm ready. So if at all you want to resemble them, I'll just use some this is a blend this is a clay made up of waste blended molded dough, like from different colors. Whatever is left out, I just mix them forming this. So with this you can just prepare this secondary cell wall which is released from the primary cell wall. So these are the secondary cell wall substances like the lignin, suberin, and in some cases the cell wall generally releases an outer layer like cutin in case of the epidermal cells, right? So the substances that are released from the primary cell wall are known as the secondary cell wall. Generally, it is made up of substances like the lignin, suberin, etc. Okay, so now when compared to the animal cell and the plant cell, the main immediate difference you find out is that plant cell will be having a cell wall, whereas animal cell will be devoid of any cell wall. So coming to the cell membrane part, we are going to prepare the cell membrane part in the same way how you prepare the cell wall. So I'm here using a little bit blue colored dough. So after taking the blue colored dough, similarly in case of cell wall habitat, we are going to blend it like this. I'm taking it on the floor surface and I'm just going to do it like this. So I have prepared the pieces of cell membrane, and you're going to arrange these pieces of cell membrane below the secondary cell wall, and make sure that the bridges, the places between them are equal. So I have placed the cell membrane parallelly to the secondary cell wall here, and here make sure that all the three are present at the similar alignment. And you can use this pen knife or just your hand, and you can just adjust them like this. So don't mind the curvy appearance, because when you do it, you can just adjust it according to your preferences. So coming to the spaces present here, what exactly are the spaces? So see, in case of plant cells, cells are so mutually present. So in order for the transportation to occur between cell to cell, there will be bridges between the cells. So bridges is nothing but the openings present in between the cell to cell. These are known as plasmodesmata. The word desmata or the desmos generally mean the bridges. So it is continuous with the interior of the cell, interior of one cell to the interior of another cell. So the cellular transportation occurs through the plasmodesmata. So that is why we have left a space between the cell membrane, cell wall and the secondary cell walls, right? So coming to the next part, it is the cytoplasm. For the cytoplasm is the clear fluid present in between the cell, and it contains all the cellular organelles. So as I have already used blue color here, I am using yellow color in order to represent the cytoplasm. So in order to form the cytoplasm, you are going to place the dove in the middle of the cell, and you are going to spread it evenly from the middle to the exterior, and you're going to do it all over the cell in an even manner. So after filling the new cytoplasm, it somehow looked like this. And here I wanted to mention you something. See, in this Rangila molding clay, the clay is little bit, it is extremely soft, but it generally breaks when you are trying to spread it. Okay, so in such conditions, what you're supposed to do, you're just going to place it in a particular way, and little after spreading, you're going to dab it like this. The more you are going to dab it, the firmly it will be sticking to the plate. Okay, so coming to the plant cell. So till now what we have discussed, that plant cells generally have a cell wall. So that is the main difference between animal cell and the plant cell. And adjacent to the primary cell wall, you'll also have the secondary cell wall, which are nothing but the secretions that are released from the primary cell wall. So after that we have the cell membrane, and cell wall is completely permeable. It allows everything inside and everything outside of the cell. So then what is the function of cell wall? Cell wall generally gives structural support to the cell and also offers protection to the cell. Whereas the cell membrane is selectively permeable and it allows only certain substances to enter and exit the cell. And coming to the cytoplasm, it is the jelly-like fluid which is present inside the cell, and within which all the organelles will float. Now coming to the nucleus, I'm using this pink color for the nucleus, and nucleus is generally a round ball like structure. So instead of representing it in a round ball, I'm just going to use it, just represent it in a flat circle. So for that, first you just going to make a small ball like this, and after making the ball using the both hands, you just press it like this, right? So press it in an even manner and make it in the form of a circle. If you want, you can also use it, prepare it in the form of a ball, as shown in some plant cell diagrams. So in case of the animal cell, the position of the nucleus is always in the middle, but in case of the plant cell, the position of the nucleus is towards its periphery, because there's always a big vacuole in the cell wall which will be pushing the nucleus to the periphery, right? So here we have already completed preparing the nucleus. So what is present inside the nucleus? Yes, the genetic material. In order to represent the genetic material, I'm using the violet color. So the genetic material is generally present in the form of DNA. DNA, when it is condensed tightly, it will form the chromatin network. So in case of the formation for the chromatin network, I'm just wrapping it like this, and the wrapping must be as uneven as possible, because you know how the condensed DNA looks, right? So I'm preparing it like this so that it looks as uneven as possible, and we are going to just immerse this into the nucleus, right? Yes, so we'll go ahead. Right, so we are going to place it in an uneven manner. And you can also make it as thinner as possible. So let us make it a little more thinner to make the presentation more elegant. So I'm just dabbing it over a hard surface and making it into a thin this thing. So after that I'm going to twist it. After twisting, when you are twisting, just try to press it and twist. After twisting, just in an uneven manner, you arrange the genetic material or the DNA. Right. So here's the nucleus, and inside the genetic material also is visible. What is the control center of the nucleus? Yeah, it is the nucleolus. So nucleolus is nothing but a spherical body present inside the nucleus, and it will be controlling the nucleus. Right, so this is the nucleolus, and this is the genetic material present in the form of DNA. And we all know that nucleus is a double membranous structure. It is covered by, yes, an outer membrane. So in order to represent the outer membrane and the pores in that, uh, we can just make the pink colored wrappings like this and place it a little like this, just to make sure, just to tell that the nucleus is a double membranous structure. And we also know that the nucleus is generally have the nuclear envelope will be having the, yeah, nuclear pores. So that's why the nuclear envelope will not be continuous. So you can just represent it like this with breaks or the spaces in between, and you can just make these spaces little more visible with the help of, see, any kind of adjustments. Instead of doing with your own hand, you can use this knife, right? Like this, you're going to separate the nuclear envelope, which shows that the nucleus is having pores. So here is the nuclear envelope with the nuclear pores around it. And now coming to the vacuole. Vacuole is the largest part of the plant cell, where it occupies majority portion of the cell. So in order to represent the vacuole, I'm just using the blue color, because like vacuole is generally filled with the portion of fluid, and generally people say that vacuoles are the storehouses of the cell. See, I just prepared a very big round, and I'm just dabbing it and making it little bit bigger. Right, so we were discussing that vacuoles are the storehouses of the cell, but apart from, you have to know that apart from storing, they also have a function, have the functions of, digesting any waste kinds of proteins, or waste any waste organelles or any damaged organelles and all. So the vacuole is generally already prepared, but you can't place it like this because it gives a very worse look, I'm not mind my language, I was not able to, I meant to say that it looks a little bit odd. So you can just take small scissors and give it the shape of a beautiful vacuole. But make sure that the vacuole is big enough and you're not making it very small. And I'm just cutting it like this in order to give it a proper shape, right? Make sure that they're just good and clean. Right, so I'm placing the vacuole now. So the large big vacuole at the center, with a little curvy manner you're going to place it, right? You're going to spread it. So after you place it down, you're not bothered about its breaking or something, so you can happily spread it to the length of how much you want. Make sure that it is not tearing at the middle, right? Right. So now after dabbing it like this, your vacuole is almost over. Yes. So this is the vacuole present, and the vacuole is generally covered by a membrane of tonoplast. So this vacuole is very important in case of a plant cell. See, this vacuole is having the fluid known as tonoplasm. Now this tonoplasm generally offers a Turgor pressure. So what is Turgor pressure? Yes, the pressure offered by the tonoplasm to the plant cell is known as Turgor pressure, and this pressure is generally exerted towards outer side of the cell. And so from here, what happens? The pressure is exerted outwards. So as a result, what happens? The plasma membrane exerts this pressure to the cell wall. So from inside the cell wall, inside the pressure is being acted upon the cell wall. So the wall pressure will be there, which is acting from outside the cell. So the wall pressure and the Turgor pressure, wall pressure from outside and the Turgor pressure from inside are going to balance it. So this is how a cell of a plant will be raised. Okay, so generally if the Turgor pressure becomes more, it results in exosmosis. If the wall pressure becomes more, it results in endosmosis. So generally in case of the summer season or something, when this Turgor pressure becomes less, you see that the plant, the leaves and all are getting wilted, because in every cell the Turgor pressure is becoming low and the water content is being sent out. So here I am just representing the tonoplast. So yes, the tonoplast is ready. So the vacuole is well covered with the tonoplast. Now we'll go with the endoplasmic reticulum. So endoplasmic reticulum is the main transportation center, and it is continuous with the nucleus, and all the ribosomes that are formed from inside of the nucleus are escaped through the nuclear envelope, and they enter the rough endoplasmic reticulum. So in order to show you the rough endoplasmic reticulum, I'm using red color. So as the space in my presentation is little less, I'm using a very tiny and very small strands of endoplasmic reticulum, and so endoplasmic reticulum generally has an empty lumen, that means empty space within it, and it is continuous attached and it is attached to the nuclear envelope. So I'm placing the endoplasmic reticulum like this, and extending from the nuclear envelope, it extends till the, yes, it extends till the plasma membrane. So you're going to just extend this like this. Make sure that you're keeping enough lumen in it and connect it to the plasma membrane like this. You're going to close it from the other side as well now. So after placing the endoplasmic reticulum towards one side, it looks like this. Even to make your presentation more elegant, you can add one more endoplasmic reticulum here as well. So here is the final picture of our endoplasmic reticulum. So generally there are two types of endoplasmic reticulum. One is the rough one, other one is the smooth one. So whatever type of endoplasmic reticulum it may be, so in between you'll be generally finding the empty space, which is the lumen through which the transportation of the substances from the nuclear envelope happens to the cell membrane, right? And so in order to start them with the ribosomes, we are using a brighter yellow color because it will be clearly seen on the red color. So for that we are taking a small ball of yellow and you're going to make it into small tiny balls where you can embed them beautifully, and make sure that you're embedding on embedding very small part, because we have already used our nucleus, our cytoplasm in an yellow color. So I'm using very tiny pieces of yellow in order to represent the RER, that is the rough endoplasmic reticulum. I'm concentrating more on the parts where more red color is present, else chances are there that you may not spot your rough endoplasmic reticulum. Yeah, you may use some different color other than yellow, so that doesn't give you any problem. So we are almost towards the end of the video, and so after successfully placing as many RER as you want, sorry, as many ribosomes as you want, you can just skip to the formation, you can just skip to the smooth endoplasmic reticulum part. So I have just changed the color of ribosomes and kept it as white, so it will be a little bit more elegant. So now moving ahead to make the smooth endoplasmic reticulum, we are going to use the same red color. So with the smooth endoplasmic reticulum, I have made a tiny strand of red color, and we are going to place it in the cytoplasm. And you're just going to join them. The smooth endoplasmic reticulum gets their name because they're devoid of ribosomes, so their appearance is generally little bit smoother when compared to the rough endoplasmic reticulum. In case of RER, the roughness is because of the presence of studded ribosomes. So if you want, you can make other tiny smooth ER wherever you want in the cytoplasm, because they are generally randomly distributed, right? So I have placed two SER. So almost all parts are finished. Now we are going ahead to the lysosomes part. In case of the for the lysosomes part, I'm using orange colored balls and pressing it like this. So two lysosomes are more than enough, and lysosomes are generally filled with fluid-filled enzymes. So I'm just pointing those in that filled liquid like this, right? So these are lysosomes. And next, coming to the other part, in case of plant cells, centrioles are absent. Centrioles generally appear during the cell division near the nucleus in order to, separate the genetic material. But in case of plant cells, our centrioles are absent, and so instead, phragmoplast, a cell plate, will be dividing the total cells. So you have observed the missing nucleolus here. Yeah, I know. So I just wanted to keep another bright color here. So I'm going with the green itself. Um, yeah, I just made it around, and I'm just dabbing it like this. Yes, right. So now we are stepping towards the plastids. Plastids are generally double membranous structures and are of three types. So in order to make the chloroplast, I'm just taking this round ball and just observe it carefully. I'm just making it into a spherical structure and then little dabbing it Now I'm using the nail cutter, so the knife part, and I'm just removing a part of chloroplast here, right? So chloroplast generally contains the green colored chlorophyll pigment, because of which the plant cell gets its ability to trap the sunlight. Now this sunlight is converted to the chemical energy, and that's why we call plants as the primary producers. So it is looking like a basic shoe type, right? So now here, as the chloroplast generally are the bodies which are rod shaped bodies. Now inside the chloroplast you will be having the chlorophyll pigments. So you also observe the grana and stroma in case of chloroplast. So the grana are arranged in the form of stacks known as thylakoids. So in order to represent the thylakoids here, you just need to take small amount of green this thing, dab, and you're making it small balls, dab it. So this is one grana, right? And keep it here. Take another ball like that similarly, dab it over this again, and make such grana. So that gives you the appearance of, yeah, like the thylakoids are present in case of the chloroplast. So that is how we made the chloroplast. And next, coming to the mitochondria. In order to prepare mitochondria, I'm using the white ball because that's the only color which is less used here. So I'm just taking it a ball, and I'm just making it into a small tiny rod, and I'm going to dab it. I'm going to place it as it is here, and you all know what we are going to do next. Yes, we are going to place the second innermost membrane of the mitochondria, which will be folded inwards like this, right? So the second membrane, the outer membrane of the mitochondria is normal, but the second membrane of the mitochondria is folded inwards. So these foldings are generally known as cristae. You know what, we also see the DNA in case of mitochondria, but these are non-chromosomal DNA, right? So we are going to complete the inner membrane. So inner membrane is like this, and it is folded occasionally, and these foldings are known as cristae, right? So this is mitochondria. So if the mitochondria is not visible here, right, so you can just dab this surface and make it little bigger, okay? So like, so finally this is how your plant cell is going to look. We have placed the chloroplast, we have placed mitochondria, lysosomes, vacuole, nucleus, ear, smooth ear, and everything. So occasionally in the cytoplasm, we also see the polysomes, which are nothing but the group of free ribosomes that are floating in the cytoplasm. So in order to represent the polysomes, I just took white balls and I'm placing it in a group here, and yeah, like this. And you know that in case of any cell, there will be number of cell inclusions, which are this the non-living molecules, like the lipids, fats, carbohydrates, and all. And apart from this, you can also include the peroxisomes. The peroxisomes are just nothing but the organelles which are involved in the oxidation of the, you know they play a key role in the oxidation of specific biomolecules. Such peroxisomes can be represented here and there, right? And generally, as for the representation purpose, I'm doing a small one. You can just do bigger ones in case of your presentations, right? So ending towards the ending, your plant cell looks like this. Thank you.